So in today's video we are going to discuss about mitochondria. Mitochondria, as we all know from lower classes, mitochondria is also known as powerhouse of cell. Okay, powerhouse or power converting organelle, and it is also known as power plant, power plant, powerhouse, or power converting organelle. Why we are giving this name? Because mitochondria is an only organelle which is capable of producing ATP. It produces ATP for the cell. What is ATP? ATP, as we all know, it is a adenosine triphosphate. It is a kind of energy currency for the cell. Okay, this ATP contains anhydride bond, phosphoanhydride bond, which when break down it will give some energy to the molecule, or you can say to the particular cell, and this energy can be utilized in any other work. So we require energy molecule, and this energy molecule is produced by mitochondria. Okay, so let us see how mitochondria looks like, and when it was discovered, and who discovered this mitochondria. Mitochondria, we all know that it is present in aerobic cell. Okay, in eukaryotes. Aerobic cell, why I am saying aerobic cell? Because those cell which requires oxygen for respiration, they will contain this organelle. Okay, so that they can utilize the hydrogen accepting molecule and can oxidize back them to the reduced form. Okay, so no, they are actually, these mitochondria will be present in organism, so that they can oxidize this NADH into NAD plus. Okay, this NADH, FADH. What are these? These are hydrogen accepting molecule. Okay, so these are further, their hydrogen is removed from here and they come back in the form of oxidized form. Okay, they are reduced during the process of glycolysis and Krebs cycle. Why I am talking about these two molecules? Because these two molecules are responsible for the formation of ATP. Okay, they are indirectly responsible for the formation of ATP. So we were talking about them. Now let us see, first of all we are going to discuss the structure, and then we will understand that what are these molecules and from where they are coming. Okay, so first of all, mitochondria was discovered by Rudolf Kölliker in 1880. In 1880, Rudolf Kölliker was the man to discover mitochondria. Okay, and he was saying that this is secule. The word he used for mitochondria was secule. In 1897, the next one, that is your Benda, he came and he gave the name mitochondria. Okay, mitochondria, this term was coined by him in 1897. Now this mitochondria is actually a fluid filled cavity. Okay, it is actually a cylindrical shaped, or you can say spherical shaped structure, and this structure is double membrane. Okay, it is enveloped by double membrane. It contains two membrane. inner membrane as well as outer membrane. Now let us see where is inner membrane. This inside, here it is inner membrane, and here outer side it is outer membrane. Okay, so it contains two membrane, outer membrane, inner membrane, and each membrane is around 45 to 75 Armstrong thick. Okay, but your outer membrane is relatively thin in compared to inner membrane, as it contains lesser protein. Outer membrane contains lesser protein as well as cardiolipin, that is a kind of phospholipid which is present in lesser amount in outer membrane but more in inner membrane. Now inner membrane also contain the protein carrier molecules. Okay, it contains certain enzymes that is required for different process which occurs in mitochondria. The liquid or fluid that is filled in mitochondria, it is known as matrix. Like cell contains cytoplasm, in same way mitochondria contains matrix. Okay, now this involution or this kind of process you are seeing inside, these are the infolding of inner membrane. The inner membrane is not straight like this, but it is a infolded. There is a certain partition, folding like structure. Okay, you can see this is the inner membrane folded in the form of partition, incomplete partition, and this partition is known as cristae. The word used for this is cristae. Okay, so here mitochondria contains cristae. There are many cristae present inside the mitochondria. Inner membrane forms many infolding, continuous infoldings, involution, which is known as cristae. Why there are foldings inside? Because we want to increase the surface area. To increase the area, surface area for activity. Okay, what kind of activity they want to do? Actually, they want to accommodate lots and lots of protein. So they will need a larger area, and for this larger area they are going to infold all the inner membrane. Inner membrane will get folded so that there is a larger area, for more and more protein can be accommodated, more and more protein can be located on this inner membrane. Okay, so one more important point, on this cristae there are oxysome. See this bulb like structure, knob like structure, this is oxysome, which is also known as elementary particle. Elementary particle, and this elementary particle is also known as F0, F1 particle. Okay, so this was the structure of mitochondria. Let us see the function. There are several process like Krebs cycle. Krebs cycle is also known as TCA, that is tricarboxylic acid cycle, and it is also known as citric acid cycle. All this cycle will occur in they are same. Okay, TCA, citric acid cycle, Krebs cycle. Now this will occur in mitochondria, in the matrix of mitochondria. Okay, it will occur in matrix of mitochondria. Okay, then the another process, that is your electron transport system, your electron transport chain. This is a process where the electron is transferred from those energy molecule, what I was saying about NADH, FADH. Okay, these are hydrogen accepting molecule which were formed during the process of Krebs cycle. And one more, there is a process glycolysis. Glycolysis is breakdown of sugar which occurs in cytoplasm, breakdown of glucose which occurs in cytoplasm of cell. So both these process will form this NADH and FADH. Okay, so these are the hydrogen accepting molecule formed during this process, Krebs cycle and glycolysis. Now these are further oxidized, they are converted into the form of NAD plus and FAD plus. This will happen during electron transport system, okay, electron transport chain. So during this process, it is hydrogen is removed from NADH and it is transferred, hydrogen is ionized in the form of proton and it is transferred from matrix to the intermembrane space. What is intermembrane space? It is a space between outer membrane and inner membrane. The space between the outer membrane and inner membrane is known as intermembrane space. So now this intermembrane space is filled with proton. From where these protons are coming? These protons are coming from NADH, FADH. Okay, when this NADH or FADH is oxidized into NAD plus and FAD plus, that time this particular proton is transferred to the intermembrane space. Why they are doing this? Because they want to generate proton gradient. Okay, they want to create the difference between the proton concentration over here in intermembrane space and in matrix. Okay, so this happen, lots and lots of H plus will get gathered over here in your membrane space. Okay, and they will come back, all the proton will come back, they will move back to matrix. But how? When they have to produce ATP. Okay, ATP is produced by process of oxidative phosphorylation. This oxidative phosphorylation will require proton gradient. Okay, this oxysome, this is your oxysome. Okay, this is your F1, this is your F0. Okay, and it is located on inner membrane space. Now this is inner membrane of mitochondria. This is your intermembrane space. Okay, now this space contains lots of proton. This proton will move through this F0 F1 complex, that is your oxysome. It is also known as ATP synthase. When it will move from here to here, that time there is a rotation of this oxysome which will cause conformational change, which will help in formation of ATP. ठीक है, ATP, formation of ATP will occur. There is an addition of phosphate on ATP. Okay, due to transfer of proton from space to matrix, and there is a formation of ATP. Okay, so this whole process, your Krebs cycle, glycolysis, ETS, that is electron transport chain, all will occur for what? For only particular aim, that is production of ATP. And where this ATP is getting produced? They are getting produced in mitochondria. Okay, location is oxysome. Okay, so mitochondria is energy providing complex, or it contains energy providing complex. It is powerhouse of cell, power plant. Why we are saying like that? Because it will produce ATP through oxysome which is present on its cristae. Other than this, mitochondria, one more specialty is there, that it is a kind of semi-autonomous organelle. Okay, we are saying autonomous organelle. Okay, it is independent organelle. Why we are saying independent? Because it contains its own DNA, double-stranded DNA. It contains its own double-stranded DNA in it, and this double-stranded DNA can either occur in form of circular or in form of linear. Generally it is circular DNA like prokaryote. Then it will contain same way in prokaryote, as prokaryote contains 70s ribosome, in same way mitochondria also contain its ribosome, 70s ribosome. Okay, so it is capable of making its own DNA, its own RNA of all the three types, mRNA, tRNA, rRNA. Okay, so therefore it is concluded that it is a autonomous, semi-autonomous organelle. Okay, so semi-autonomous, why we are using word semi? Because it will not survive like this out of the cell. It wants cell, it requires cell for its survival. So therefore it is semi-autonomous, but it will replicate its own DNA, it will cause its own division, everything. Okay, so it is a kind of semi-autonomous organelle. So this was about mitochondria, and I have tried to cover all the point related to mitochondria. Thank you.